Okay, so the next speaker is Francesco Gammaraccio. Okay, and I will talk about the software that we are developing in order to expand some of the capabilities of XNAT. Please, Francesco.
So, good morning, everyone. My name is Francesco Gammaraccio, and today I will present you the tools that we have developed to expand the XNAT core functionality to preclinical imaging centers. So the point that we will discuss today are an overview of preclinical imaging. So we will see a brief introduction, the challenges, the first principles, and the current preclinical images archives. Then I will present our desktop application called XNAT-PIC, that stands for XNAT for Preclinical Imaging Centers. So preclinical imaging refers to the use of different types of imaging techniques to study biological structures and activities in small animals, such as mice, rats, zebrafish, and so on. And the imaging techniques that are common used in preclinical research are CT, MRI, and so on. So it's clear that the preclinical centers generates a large amount of data and information, but there are not many common applications for storing and sharing preclinical imaging datasets. And then preclinical research addresses specific scientific questions. For example, if we want to monitor the effect of a therapy, we need different groups, for example, treated and untreated. And the studies are usually longitudinal. It means that we want to study the effect over the time. So we need to acquire images over the time. For example, for time t equal to 0 or t equal to 1 and so on. And then we want to study the drugs effect. So we don't have a standard protocol, but for each specific question, we have a specific protocol. And then another challenge is related to the proprietary raw image, because the images are stored in proprietary file format, and so they depend on the vendor, the software and the hardware of the scans, and they depend on the version of the software installed. And then for clinical imaging, we have the DECOM standard, that is the worldwide recognized format for clinical images. Whereas at preclinical level, we have the full information saved on the raw data, raw files, and the DICOM doesn't have the full information. So at preclinical level, DICOM standard needs updates for emerging imaging technologies, such as the chemical exchange saturation transfer imaging. At preclinical level, the images are stored in local databases or in local PC, so they are not findable, and of course, they are not accessible from outside. And from the point of view of the interoperability and the reusability, information and metadata are not common defined. So preclinical center can use specific vocabularies, terms, but they are not fully recognized by others in the same or in others field. At preclinical level, we have different archives for storing and sharing images. For example, we can have in-house databases or platform that the IET staff can develop. And then we have Chenoir, that is a web platform developed at French national level for clinical and preclinical research, for importing, sharing, searching, and visualizing medical imaging data. And there is Flywheel, that is a commercial data management system. So we don't have a solution for our needs, so we decided to use XNAT for preclinical imaging center. But why to choose XNAT? XNAT is an open source platform, and it works as a database. And XNAT is a imaging platform for managing and sharing imaging dataset. And we have the possibility to develop and use and share processing tools. And XNAT supports the FAIR principle. But the problems are that XNAT is not optimized for importing structured preclinical imaging datasets, and XNAT is not able to manage common custom variables such as groups, time point, doses used in preclinical studies. So our aim is to provide an easy-to-use desktop application for managing and annotating preclinical image datasets. And we want to overcome the limitations of XNAT for preclinical imaging. So we have developed XNAT-PIC, that stands for XNAT for Preclinical Imaging Centers. That is a free and open source Windows desktop application. The features that XNAT-PIC offers are a grouping annotation interface, that is a dedicated interface to edit the custom variables for your project. So this interface allows you to select your project, and you can edit the custom variables that you want to add, such as groups, time point, doses, and so on. And then XNAT-PIC offers a converter to convert Bruker file format to DICOM standard, and we have added the possibility to save some private DICOM tags for new imaging technique, such as the CES imaging. And then XNAT-PIC allows you to import image dataset into the XNAT platform in a very easy way. And the uploader supports the entire project, sessions, subject, experiment. So talking about some technical details, XNAT-PIC was developed in Python. We have used different packages, but the main are XNAT-PI, that allows you to communicate with XNAT. We have used PyDICOM to save the DICOM images, and then NumPy for the converter. And then we have used a library to save credential with a cryptographic algorithm. XNAT is an open source desktop application that is distributed under the generic public license. So the first version of XNAT-PIC was developed by Sarah Zullino and colleagues. The main features that it provides are a converter for experiments, and you can also upload your experiment to the XNAT platform, and the custom variables are edited based on the folder structure. But now we have developed a new version of XNAT-PIC, the version number 2, that has new updates. XNAT-PIC now is a standalone software, and we have designed a user-friendly interface. We have secure access, and we have improved the speed of the converter. We have introduced an interface for grouping annotation, and now the converter for Paravision 316 is supported, and the user can upload the entire project, subject, experiment, and also non-DICOM file. So XNAT-PIC is very easy to install on your PC. You only need to download the installer file from our website. Here we have the QR code, and we also provide the source code on our GitHub link. Here there is the link. We have designed XNAT-PIC with a user-friendly interface inspired by Bootstrap. This picture shows you the main interface where we have three options: the DICOM converter, the button to open the dedicated interface for custom variables, and the uploader. The credentials, such as the IP, the username, and the password to connect to an XNAT instance, are protected by a cryptographic algorithm. The figure on the right shows you our XNAT instance called Euro-BioImaging. And we have added multi-core processing for improving the speed of the converter. For example, for one project that contains 42 subjects and 42 experiments that are more or less 650 MRS scans, it takes 6 minutes to convert the entire project. So preclinical studies address specific scientific questions. So we can have different groups because we want to monitor the effect of the therapy, and we want to compare the different groups. So we can have group treated and untreated, and we also have different time points and different doses. And the folder structure on the left shows you this grouping. And if we upload this project to XNAT, we cannot see the custom variables. So we have the XNAT-PIC interface for custom variables to facilitate the grouping annotation. In this case, we have selected a project and we have added some custom variables. And then we have added again, we have uploaded again the project to XNAT, and now we can see the custom variables for the subjects. And XNAT-PIC version 2 supports the converter for Paravision 316, and we have added some private tag for emerging MRI techniques, and the picture shows you the private tags. And then the user has the possibility to upload the entire project, subject, and experiment to XNAT and also non-DICOM files. For example, you can have a folder that contains parametric maps, TXT files for your results, and you can upload to XNAT. The picture here on the right shows you this folder with the parametric map and the results TXT file. So to sum up, XNAT is a powerful imaging platform, but it is not optimized for preclinical studies. So we have developed XNAT-PIC, that stands for XNAT for Preclinical Imaging Centers, and it has some features such as grouping annotation interface, the converter, and the uploader. And this is okay. Thank you for the attention.